Hi everyone, my name is Malik, and I am one of the multi-organ team leads at Hespros, which is a human on a chip company. Today I'll be talking about some of the very interesting work that we are doing at Hespros, which is utilizing an in vitro multi-organ platform to look at some of the safety and efficacy of some novel therapeutics. One of the models that we have developed in-house is a gastrointestinal, liver, blood-brain barrier, and CNS model. So we have four different organ constructs within a single platform that is gravity-based. So we have gravity-driven flow across the four constructs. We have a representation of the device within this poster. The project was done in collaboration with Bayer Pharmaceuticals, where we looked at the potential of using Echinacea purpura, which is an herbal extract, to look at its potential activity on relieving stress. So Echinacea purpura has an active ingredient, which is Dodeca. So within the system, what we started to target was utilizing a stress CNS. So the CNS or the cortical neurons were induced chemically with cortisol to inject a stress phenotype within the CNS. And this particular model has been published previously by our group to establish that stress phenotype. The advantage of this multi-organ platform is with the different modularities that it has, we can dose the drugs using different routes. Since echinacea is generally prescribed as an immunomodulator currently, that is what it is well known for, it is prescribed as an oral tincture. So within this organ construct, to replicate or mimic that oral delivery, the drug was dosed directly into the GI, across which it transported, then distributed within the blood path to interact with the liver, BBB, and eventually CNS. Within this microphysiological system, multi-organ platform, we not only dosed the system with echinacea, but also looked at five different concentrations of pure dodeca, which is the active ingredient within echinacea. CBD and diazepam, which are two well-known stress and anxiety relievers, were also included to serve as positive controls. Interesting data that we found through this study was looking at endpoint viability and general maintenance of the four organ constructs over a 48-hour exposure period. So we did have a single dose, but monitored the system for 48 hours to look at the different responses of the different constructs to the drugs. What we observed was that the viability of the four different constructs was maintained more or less across the drug concentrations. Additionally, with the BBB and GI, we looked at tear maintenance, which is an indicator of barrier integrity. Looking at that, we saw that with the exposure of the GI to the echinacea extract, we did see the tear go up at the 48-hour time point, indicating the rescue effects, more or less, that the echinacea shows to the barrier integrity. There were no significant losses that were seen during the first 24 hours. At 48 hours with the BBB, we did see a dose-dependent reduction in tear from the threshold that we had set. However, when we look at some stress biomarkers, so we looked at anandamide and 2-HG, we saw that the free anandamide concentration was increased with increasing concentration of dodeca, which is the pure dodeca that we utilized in this study. This indicated that free anandamide, which recognizes the same target as dodeca, which is CB1 receptor, was widely available within the construct, which means this construct, which is the CNS, which is stress induced, was experiencing reduction in stress. Liver health was also maintained, no toxicity was seen. CYP1A1 and 3A4 activity, which the two CYP enzymes that are majority of the time involved in breakdown of drugs, was seen to be maintained, if not enhanced or increased with the use of extract. The two positive controls, diazepam and CBD, they have varying mechanism of action to relieve stress. while we saw diazepam working similar to the extract by inducing 1A1 activity, we saw that CBD, in fact, was able to rescue long-term potentiation, which is a functional activity measurement of the cortical neurons. So over the course of 48 hours, we evaluated the changes in LTP in response to the drugs and saw that when the CNS was intrathecally dosed as a single organ platform, we saw a significant increase in LTP at the 4-hour time point with the extract, which was seen to be fleeting and went away by 24 hours. On the other hand, within a multi-organ platform, we saw the same fleeting effect, except now with the inclusion of liver, which contributes to the metabolism, and then also having the two different barrier platforms, which is the blood-brain barrier and the GI, we now introduce pharmacokinetics. So these two physiologically relevant phenomena contributed to the effect that we see within the multi-organ platform. We still saw that fleeting effect that I was mentioning with the extract. So we saw a tiny spike at the 24-hour LTP maintenance within the multi-organ, which went away by 48 hours. Interestingly, CBD, which again is known as a stress reliever, we saw an enhanced LTP maintenance at all the 3 time points within the multi-organ platform, reiterating what we see in literature where CBD is not only contributing to that stress relieving by itself, but also its metabolites, which are being produced in presence of the liver in our multi-organ platform. Another interesting observation we made was echinacea extract, which initial hypothesis said that dodeca, one of the most active ingredients within echinacea, is primarily involved in this rescue effect. However, when we compared the PKPD profile of the pure drug, which is the pure dodeca, versus the herbal extract, we saw that at the similar concentration, so echinacea extract had an equivalent concentration to 1 micromolar of dodeca, which is the lowest concentration we use in the study. The extract showed an effect on LTP, which was comparable to 167 micromolar of dodeca, showcasing that it's not just dodeca that is contributing to this effect, but an amalgamation of other components within the herbal extract. Utilizing this multi-organ platform, we not only saw no potential toxicity to the other constructs tested, but in fact saw that the inclusion of liver and these barrier platforms compares more to the physiological relevance as opposed to using some single organ constructs. In totality, this particular multi-organ platform can also be extended to include other organs, such as the cardiac platform, to look at functional responses and potential secondary toxicity. So this opens not just a door pertaining to echinacea, but other compounds as well to look at the safety of those drugs. So I hope you found this talk interesting, and if you have any further questions, please feel free to stop by my poster, and thank you for attending.